Question 1. Comparative studies suggest that in a regulated environment for a specialist in microbiology, SM, interpreting complex transcriptomic and metabolomic data requires a nuanced approach. Given a dataset showing differential gene expression and corresponding changes in metabolite profiles in a microbial culture under investigation, which of the following represents the most scientifically rigorous and regulatorily compliant interpretation strategy? Option A. Correlating observed metabolite changes with specific gene expression patterns and validating these correlations against established biochemical pathways and existing scientific literature to build a mechanistic understanding. Option B. Prioritizing the interpretation of the most abundant metabolites detected without considering the underlying gene expression that might have led to their production. Option C. Focusing exclusively on the genes that show the most significant upregulation in the transcriptomic data, assuming these directly drive all observed metabolic shifts. Option D. Drawing conclusions based on preliminary, unvalidated correlations between individual gene expression changes and metabolite levels, relying on anecdotal observations to support the findings. The correct answer is A. Correlating observed metabolite changes with specific gene expression patterns and validating these correlations against established biochemical pathways and existing scientific literature to build a mechanistic understanding. Explanation. Scenario analysis. This scenario presents a professional challenge in interpreting complex omics data, transcriptomics and metabolomics, within the context of a regulated environment for a specialist in microbiology, SM. The challenge lies in translating raw, high-dimensional biological data into actionable insights that comply with regulatory expectations for accuracy, reproducibility, and scientific rigor. The SM must make a critical decision regarding the interpretation and reporting of findings, knowing that misinterpretation could lead to incorrect conclusions about microbial function, potential safety concerns, or flawed product development, all of which have regulatory implications. The need for a robust, evidence-based approach is paramount, especially when dealing with novel or complex biological phenomena. Correct approach analysis. The correct approach involves a multifaceted interpretation that integrates findings from both transcriptomic and metabolomic datasets, supported by established biological pathways and literature. This approach represents best professional practice because it acknowledges the inherent limitations and complementary strengths of each omics technique. Transcriptomics reveals gene expression patterns, indicating potential metabolic activity, while metabolomics directly measures the presence and abundance of metabolites, providing a snapshot of the actual biochemical state. By correlating changes in gene expression with observed metabolic shifts, and validating these correlations against known biological pathways and existing scientific literature, the SM can build a more robust and mechanistically sound interpretation. This aligns with regulatory expectations for scientifically valid data and well-supported conclusions, ensuring that any reported findings are reliable and reproducible. The emphasis on correlation and mechanistic understanding, rather than isolated observations, is crucial for demonstrating a deep grasp of the microbial system under investigation. Incorrect approaches analysis. Focusing solely on transcriptomic data without considering metabolomic evidence represents a significant regulatory and ethical failure. Transcriptomics indicates potential, but not necessarily actual, metabolic activity. Genes may be expressed but not translated into functional proteins, or the resulting proteins may not be active under the specific experimental conditions. Relying only on this incomplete picture can lead to overestimation or misinterpretation of metabolic capabilities, potentially resulting in flawed risk assessments or product claims that are not supported by the full biological reality. Prioritizing metabolomic data exclusively while disregarding transcriptomic insights is also professionally unacceptable. While metabolomics provides direct evidence of metabolic products, it does not explain the underlying regulatory mechanisms or the genetic basis for these changes. This can lead to a descriptive but not explanatory understanding of the microbial system. Without understanding the gene expression that drives metabolic changes, it becomes difficult to predict how the system might respond to different conditions or to identify specific targets for intervention or manipulation, which is often a key requirement in regulated research. Interpreting findings based on anecdotal evidence or preliminary, Unvalidated correlations without rigorous cross-validation between the two omics datasets is a critical failure. This approach lacks scientific rigor and is highly susceptible to false positives or spurious correlations. Regulatory bodies expect interpretations to be based on robust, reproducible data and sound scientific principles. Anecdotal interpretations are inherently subjective and cannot be relied upon for critical decision-making in a regulated environment, potentially leading to noncompliance and a lack of scientific credibility. Professional reasoning. Professionals in this field should adopt a systematic, integrative decision-making framework. This begins with clearly defining the research question and the expected outcomes. Next, it involves a thorough quality control assessment of both transcriptomic and metabolomic datasets. The core of the process is the integrated analysis, where potential correlations between gene expression and metabolite levels are identified. These correlations must then be critically evaluated against known biological pathways, existing literature, and experimental context. Any significant findings should be further validated through independent methods if possible. Finally, the interpretation and reporting must be clear, concise, and directly address the research question, explicitly stating the evidence supporting each conclusion and acknowledging any limitations. This structured approach ensures that decisions are data-driven, scientifically sound, and compliant with regulatory standards. Question 2. The audit findings indicate that several culture plates incubated under standard conditions for bacterial identification have shown inconsistent growth patterns, with some exhibiting overgrowth of common contaminants and others showing significantly reduced growth of expected target organisms. The laboratory operates under UK regulatory frameworks for diagnostic microbiology. Which of the following investigative approaches best addresses these findings? Option A. Immediately recalibrate all incubators to a slightly higher temperature setting, assuming that increased heat will universally improve bacterial growth and outcompete contaminants. Option B. Focus solely on implementing a more stringent media sterilization protocol, believing that the issue is primarily due to microbial contamination introduced during media preparation. Option C. Request an immediate external proficiency testing sample to assess overall laboratory performance, without conducting an internal investigation into the specific environmental factors. Option D. Systematically assess the current incubation temperature, pH of the culture media, and the oxygen levels within the incubators, correlating any deviations with the observed growth inconsistencies and the known physiological requirements of the target and contaminant microorganisms. Correct answer is D. Systematically assess the current incubation temperature, pH of the culture media, and the oxygen levels within the incubators, correlating any deviations with the observed growth inconsistencies and the known physiological requirements of the target and contaminant microorganisms. Explanation. Scenario analysis. This scenario is professionally challenging because it requires the microbiologist to interpret audit findings related to critical environmental controls for microbial growth in a regulated laboratory setting. The challenge lies in accurately identifying the root cause of the observed deviations and proposing corrective actions that are not only scientifically sound, but also compliant with the specific regulatory framework governing specialist microbiology laboratories. The pressure to maintain sterility, prevent contamination, and ensure the integrity of diagnostic results necessitates a thorough and evidence-based approach. Correct approach analysis. The correct approach involves a systematic investigation that prioritizes understanding the interplay of temperature, pH, and oxygen levels on the specific microorganisms relevant to the laboratory's work. This approach aligns with best professional practice by focusing on the scientific principles of microbial growth and their direct impact on laboratory operations. It demonstrates adherence to regulatory expectations that laboratories maintain controlled environments to prevent false positives or negatives and ensure the reliability of diagnostic testing. By considering the specific requirements of the microorganisms and the potential impact of environmental fluctuations, the microbiologist is acting proactively to address the audit findings and prevent future occurrences, thereby upholding the quality and integrity of the laboratory services. Incorrect approach is analysis. An approach that solely focuses on adjusting one parameter without considering its interaction with others is an incomplete and potentially ineffective solution. For example, Merely increasing incubation temperature without assessing its effect on pH or oxygen availability might inadvertently create an environment that favors the growth of undesirable contaminants or inhibits the growth of target organisms. This failure to conduct a comprehensive analysis constitutes a regulatory failure, as it does not adequately address the root cause of the audit findings and may lead to continued noncompliance. Another incorrect approach would be to implement changes based on general laboratory best practices without specific reference to the audit findings or the types of microorganisms being handled. This demonstrates a lack of critical thinking and a failure to tailor solutions to the specific context of the laboratory. Such an approach risks introducing new problems or failing to resolve the existing ones, leading to a breach of ethical responsibility to provide accurate and reliable diagnostic services. A further incorrect approach might involve over-reliance on external validation without internal investigation. While external validation is important, it should complement, not replace, a thorough internal assessment. Failing to conduct a detailed internal investigation into the environmental factors means the laboratory is not taking ownership of its processes and is not developing the internal expertise to prevent recurrence, which is a significant regulatory and ethical lapse. Professional reasoning. Professionals in specialist microbiology laboratories must adopt a systematic, evidence-based decision-making process. This involves 1. Thoroughly understanding the audit findings and their potential implications. 2. Consulting relevant regulatory guidelines and scientific literature pertaining to the specific microorganisms and laboratory procedures. 3. Conducting a root cause analysis that considers all contributing factors, including environmental parameters like temperature, pH, and oxygen. 4. Developing and implementing corrective and preventative actions, COPPA, that are scientifically validated and compliant with regulations. 5. Documenting all investigations, actions, and outcomes meticulously. 6. Monitoring the effectiveness of implemented changes and making further adjustments as necessary. This structured approach ensures that decisions are informed, defensible, and contribute to the overall quality and safety of laboratory operations. Question 3. To address the challenge of differentiating between a bacterial flagellum and a eukaryotic cilium when observing a sample under high-resolution microscopy, which approach best utilizes the principles of microbial cell structure and function for accurate identification? Option A. A comparative analysis focusing on the distinct ultrastructural arrangement of microtubules, 9+2 in eukaryotes vs. basal body structure in prokaryotes, and the differing mechanisms of motility they facilitate. Option B. An assessment based primarily on the overall length and thickness of the appendage, assuming longer structures are more likely to be eukaryotic. Option C. An identification based on the typical cellular environment, assuming appendages in a human tissue sample are always eukaryotic cilia. Option D. A determination solely based on the motility observed, classifying any rapidly moving appendage as a cilium and any slower moving one as a flagellum. The correct answer is A. A comparative analysis focusing on the distinct ultrastructural arrangement of microtubules, 9+2 in eukaryotes vs. a basal body structure in prokaryotes, and the differing mechanisms of motility they facilitate. Explanation. Scenario analysis. This scenario is professionally challenging because it requires a microbiologist to differentiate between two structurally similar but functionally distinct microbial cell components. Misidentification can lead to incorrect diagnostic conclusions, inappropriate treatment strategies, or flawed research outcomes, all of which have significant implications for patient care and scientific advancement. The challenge lies in applying detailed knowledge of microbial ultrastructure and the specific functions of these components in a practical, diagnostic context. Correct approach analysis. The correct approach involves a detailed comparative analysis of the ultrastructural features and known functional roles of the two cellular components. This aligns with best professional practice by ensuring accurate identification based on established scientific principles and diagnostic criteria. In a regulatory framework for a specialist in microbiology, this approach is implicitly supported by guidelines emphasizing evidence-based practice, accuracy in reporting, and adherence to recognized diagnostic methodologies. The professional responsibility is to utilize all available, validated information to arrive at the most precise identification. Incorrect approach is analysis, One incorrect approach would be to rely solely on the general shape or size of the observed structure without considering finer ultrastructural details or functional implications. This is a regulatory failure because it bypasses the requirement for thorough and accurate microscopic examination, potentially leading to misdiagnosis. Ethically, it represents a failure to exercise due diligence and professional competence. Another incorrect approach would be to make an identification based on a single, non-definitive characteristic, such as staining properties alone, without corroborating evidence from ultrastructure or function. This is a regulatory failure, as it deviates from established diagnostic protocols that often require multiple lines of evidence for definitive identification. It also fails to meet the standard of care expected of a specialist. A further incorrect approach would be to assume the component's identity based on the known prevalence of certain microbes in a particular sample type, without direct microscopic evidence. This is a regulatory failure because it introduces bias and relies on probability rather than direct observation and identification, compromising the integrity of the diagnostic process. It is an ethical failure to provide a definitive identification without sufficient supporting data. Professional reasoning. Professionals should approach such challenges by first meticulously observing all available microscopic features, including size, shape, internal structures, and any associated external features. This should be followed by a critical evaluation of the functional implications of these observed features, drawing upon established knowledge of microbial physiology and pathogenicity. If uncertainty persists, consulting reference materials, seeking a second opinion from a peer, or employing additional confirmatory techniques, e.g., specific staining, molecular methods if applicable and within scope, are essential steps. The decision-making process must prioritize accuracy, evidence-based conclusions, and adherence to established diagnostic standards to ensure patient safety and scientific validity. Question 4. The control framework reveals that a research team is developing novel CRISPR-based gene editing applications for agricultural pest control. Considering the specialist in microbiology, SM, exams focus on UK regulatory frameworks and CISI guidelines. Which of the following approaches best addresses the potential impact assessment of these applications? Option A. Conduct a comprehensive risk-benefit analysis, consulting relevant UK regulatory bodies for guidance on containment, environmental release, and public health implications, and ensuring all proposed applications align with the UK's biosafety legislation and ethical guidelines for genetic modification. Option B. Focus primarily on the scientific novelty and potential economic advantages of the CRISPR applications, with minimal consideration for potential ecological or health risks. Option C. Rely on general international best practices for gene editing research, without specifically verifying their applicability or legal standing within the UK regulatory environment. Option D. Prioritize the rapid development and deployment of the technology to address immediate agricultural needs, assuming that any potential negative impacts will be minor and manageable through future adjustments. The correct answer is A. Conduct a comprehensive risk-benefit analysis, consulting relevant UK regulatory bodies for guidance on containment, environmental release, and public health implications, and ensuring all proposed applications align with the UK's biosafety legislation and ethical guidelines for genetic modification. Explanation. This scenario is professionally challenging due to the rapid advancement of CRISPR and gene editing technologies, which outpace the development of comprehensive regulatory frameworks. Professionals must exercise careful judgment to balance innovation with public safety and ethical considerations, particularly when assessing the potential impact of these technologies on human health and the environment. The specialist in microbiology, SM, exam requires adherence to specific UK regulatory frameworks and CISI guidelines, meaning any assessment must be grounded in these particular legal and ethical standards. The correct approach represents best professional practice because it prioritizes a thorough, evidence-based impact assessment that considers both intended and unintended consequences of CRISPR applications. This aligns with the precautionary principle often embedded in UK biosafety regulations, which mandates a cautious approach when scientific certainty is lacking regarding potential harm. It also reflects the ethical imperative to protect public health and the environment, as guided by principles of responsible innovation and scientific integrity emphasized in CISI guidelines. This approach involves consulting relevant UK regulatory bodies, such as the Health and Safety Executive, HSE, and the Human Fertilization and Embryology Authority, HFE, where applicable, to ensure compliance with existing legislation and to inform future policy development. An incorrect approach that focuses solely on the scientific novelty of CRISPR technology without a robust impact assessment fails to meet regulatory requirements. This is a significant ethical failure as it disregards potential risks to human health and the environment, contravening the duty of care expected of professionals. Such an approach could lead to the unauthorized release of genetically modified organisms or the application of gene editing in ways that are not ethically sanctioned or legally permissible under UK law. Another incorrect approach that relies on anecdotal evidence or unsubstantiated claims about the benefits of CRISPR technology, while downplaying potential risks, is also professionally unacceptable. This demonstrates a lack of scientific rigor and a failure to adhere to the evidence-based decision-making principles mandated by regulatory bodies. It can lead to misinformed policy decisions and public distrust, violating ethical obligations to transparency and scientific accuracy. A further incorrect approach that ignores the specific UK regulatory landscape and instead adopts general international guidelines without verifying their applicability or legal standing within the UK jurisdiction is a critical failure. This demonstrates a lack of due diligence and can result in non-compliance with specific UK laws and ethical codes, potentially leading to legal repercussions and reputational damage. Professionals must always operate within the defined legal and ethical boundaries of the jurisdiction in which they are practicing. The professional decision-making process for similar situations should involve a systematic evaluation of the technology's potential benefits against its risks, grounded in scientific evidence, and aligned with the specific regulatory framework of the jurisdiction. This includes identifying all relevant UK legislation, guidelines, and ethical codes, consulting with regulatory experts, engaging in stakeholder consultation where appropriate, and documenting the assessment process thoroughly. A commitment to ongoing monitoring and reevaluation as the technology evolves is also crucial. Question 5. Operational review demonstrates that a novel bacterial isolate from a patient with a severe, rapidly progressing infection exhibits unusual phenotypic characteristics. To definitively identify the organism and assess potential virulence factors and antimicrobial resistance mechanisms, the laboratory is considering various genomic sequencing techniques. Which of the following approaches would best ensure the generation of a high-quality, comprehensive genome sequence suitable for accurate identification and characterization in this critical clinical scenario? Option A. Selecting the sequencing technology with the lowest upfront cost to optimize laboratory resource allocation. Option B. A hybrid sequencing approach combining long-read and short-read technologies to achieve both contiguous assembly and high base pair accuracy. Option C. Solely employing short-read sequencing technology due to its established accuracy for single nucleotide polymorphism detection. Option D. Utilizing only long-read sequencing technology to obtain a highly contiguous genome assembly, prioritizing structural integrity. The correct answer is B. A hybrid sequencing approach combining long-read and short-read technologies to achieve both contiguous assembly and high base pair accuracy. Explanation. Scenario analysis. This scenario presents a professional challenge due to the critical need to accurately identify and characterize a novel microbial pathogen. The implications of misidentification or incomplete characterization can range from ineffective treatment strategies and public health crises to regulatory noncompliance and reputational damage for the laboratory. The pressure to deliver timely and reliable results, coupled with the inherent complexities of genomic sequencing, necessitates a rigorous and well-justified approach. The choice of genomic sequencing technique directly impacts the quality, depth, and cost of the data obtained, making the decision a significant one with far-reaching consequences. Correct approach analysis. The correct approach involves utilizing a hybrid sequencing strategy that combines long-read and short-read technologies. This represents best professional practice because it leverages the strengths of each technology to overcome their individual limitations. Long-read sequencing, e.g., PacBio, Oxford Nanopore, excels at spanning repetitive regions and resolving complex genomic structures, providing a more complete and contiguous genome assembly. Short-read sequencing, e.g., Illumina, offers higher accuracy and lower error rates, crucial for variant calling and precise gene identification. By integrating data from both, a highly accurate, complete, and well-annotated genome can be generated. This comprehensive data is essential for accurate pathogen identification, understanding its virulence factors, and developing targeted diagnostic and therapeutic interventions, aligning with the principles of good laboratory practice and public health surveillance mandated by regulatory frameworks for specialist microbiologists. Incorrect approaches analysis, relying solely on short-read sequencing, would be a significant regulatory and ethical failure. While cost-effective and accurate for many applications, short reads struggle with highly repetitive regions and complex structural variations, potentially leading to fragmented or incomplete genome assemblies. This could result in missed critical genetic elements, such as mobile resistance genes or virulence factors, leading to misdiagnosis or an incomplete understanding of the pathogen's biology. Such an outcome would contravene the expectation of thoroughness and accuracy required for clinical and public health microbiology. Employing only long-read sequencing, while providing contiguous assemblies, can be more prone to base-level errors and may require more extensive bioinformatic correction. If not adequately validated and corrected, this could lead to the introduction of spurious genetic variations or misinterpretation of gene sequences, impacting downstream analysis and potentially leading to incorrect conclusions about the pathogen's characteristics. This falls short of the accuracy standards expected for definitive identification and characterization. Using a sequencing technique based solely on the lowest initial cost, without considering the depth of information required for accurate identification and characterization, would be a critical professional and ethical lapse. Regulatory bodies expect laboratories to employ appropriate methodologies that ensure the reliability and validity of results, even if it incurs a higher upfront cost. Prioritizing cost over scientific rigor can lead to suboptimal data, delayed or incorrect diagnoses, and ultimately, compromised patient care and public health outcomes, violating the duty of care and professional responsibility. Professional reasoning. Professionals in specialist microbiology must adopt a decision-making process that prioritizes scientific validity, accuracy, and completeness of data, balanced with resource considerations. This involves 1. Clearly defining the objective of the sequencing, e.g., identification, strain typing, resistance gene detection, outbreak investigation. 2. Evaluating the strengths and weaknesses of available sequencing technologies in relation to the objective. 3. Considering the potential impact of incomplete or inaccurate data on clinical decisions, public health, and regulatory compliance. 4. Selecting the most appropriate methodology, which may involve a combination of techniques, to achieve the desired level of confidence and detail. 5. Ensuring robust bioinformatic analysis and validation of results. This systematic approach ensures that the chosen method is scientifically sound, ethically defensible, and meets regulatory expectations for high-quality microbiological diagnostics. Question 6. The efficiency study reveals a significant decline in overall metabolic output and biomass production in a eukaryotic microbial culture. Further analysis indicates a marked reduction in ATP synthesis and a backlog in the processing of newly synthesized proteins. Considering the known functions of key organelles, which of the following interpretations best explains the observed efficiency decline? Option A. A complete absence of mitochondria, chloroplasts, and endoplasmic reticulum within the microbial cells. Option B. The observed decline is primarily due to external environmental stressors such as nutrient scarcity, with no significant internal cellular pathology. Option C. Impaired mitochondrial function leading to reduced ATP generation, coupled with endoplasmic reticulum dysfunction hindering proper protein synthesis and folding. Option D. Reduced photosynthetic activity due to suboptimal chloroplast function, impacting the primary energy source for the microbe. The correct answer is C. Impaired mitochondrial function leading to reduced ATP generation, coupled with endoplasmic reticulum dysfunction hindering proper protein synthesis and folding. Explanation. This scenario presents a professional challenge because it requires a microbiologist to interpret complex experimental data concerning organelle function and its impact on microbial efficiency, while strictly adhering to the regulatory framework for the specialist in microbiology (SM) exam. The challenge lies in discerning the most accurate interpretation of the observed efficiency changes based on the known roles of mitochondria, chloroplasts, and the endoplasmic reticulum in eukaryotic microbial physiology, and then aligning this interpretation with the specific requirements and expected depth of analysis for the SM exam, which emphasizes a thorough understanding of microbial biology within a defined regulatory context. Correct approach represents best professional practice because it demonstrates a comprehensive understanding of how disruptions or alterations in key eukaryotic organelles directly influence metabolic efficiency. Specifically, it correctly links the observed decrease in energy production and synthesis of essential biomolecules to the impaired function of mitochondria energy generation, and the endoplasmic reticulum, protein synthesis and folding, respectively. This aligns with the SM exam's focus on detailed knowledge of microbial cellular structures and their functional significance, ensuring that the interpretation is scientifically sound and directly addresses the observed phenomena. Adherence to the SM exam's regulatory framework means prioritizing accurate biological interpretation over speculative or incomplete explanations. An incorrect approach that focuses solely on chloroplasts would be a regulatory and ethical failure because it ignores the primary roles of mitochondria and the ER in overall cellular energy and synthesis, which are demonstrably affected by the efficiency study's findings. This demonstrates a lack of comprehensive understanding of eukaryotic microbial metabolism and a failure to apply the full scope of relevant biological knowledge as expected by the SM exam. Another incorrect approach that attributes the efficiency decline to external environmental factors without considering the internal organelle dysfunction would be a failure to meet the exam's analytical depth requirements. It bypasses the core biological mechanisms at play, which is a significant deviation from the expected scientific rigor. This suggests an inability to connect observed outcomes to underlying cellular processes, a critical skill for a specialist in microbiology. A further incorrect approach that suggests a complete absence of these organelles would be a fundamental biological error and a clear failure to meet the basic knowledge standards of the SM exam. It demonstrates a gross misunderstanding of eukaryotic cell biology and would be professionally unacceptable. The professional decision-making process for similar situations involves a systematic evaluation of experimental results against established biological principles, prioritizing evidence-based conclusions. Professionals must consider all relevant cellular components and their known functions, critically assess the impact of observed changes, and ensure their interpretations are consistent with the specific knowledge domains and analytical expectations of their professional certifications or examinations, such as the Specialist in Microbiology, SM, exam. Question 7. Consider a scenario where a research team has successfully engineered a plasmid designed to enhance the bioremediation capabilities of a specific bacterial strain by enabling it to degrade a novel industrial pollutant. The plasmid contains genes that confer antibiotic resistance as a selectable marker. Before scaling up production for field trials, what is the most critical step to ensure compliance with regulatory frameworks and professional best practices concerning the potential impact of mobile genetic elements? Option A. Focus solely on optimizing the pollutant degradation efficiency of the engineered strain, as this is the primary objective of the research, and address potential gene transfer issues only if they become apparent during field trials. Option B. Immediately proceed with large-scale production and field trials, assuming the plasmid's intended function will be contained and that the antibiotic resistance marker is solely for laboratory selection. Option C. Conduct a comprehensive risk assessment evaluating the potential for horizontal gene transfer of the antibiotic resistance marker and the engineered genes to other bacteria, alongside an assessment of the plasmid's stability and the environmental impact of the modified organism, adhering strictly to national biosafety guidelines. Option D. Rely on the known stability of the plasmid in laboratory culture conditions and assume that environmental factors will not significantly alter its behavior or facilitate its transfer to other organisms. The correct answer is C. Conduct a comprehensive risk assessment evaluating the potential for horizontal gene transfer of the antibiotic resistance marker and the engineered genes to other bacteria, alongside an assessment of the plasmid's stability and the environmental impact of the modified organism, adhering strictly to national biosafety guidelines. Explanation. Scenario analysis. This scenario presents a professional challenge due to the inherent instability and potential for unintended consequences associated with manipulating mobile genetic elements like plasmids. The rapid dissemination of antibiotic resistance genes, the potential for horizontal gene transfer to other bacterial species, and the unknown long-term ecological impact necessitate a highly cautious and evidence-based approach. The challenge lies in balancing the potential benefits of engineered plasmids, e.g., for bioremediation or therapeutic applications, against the significant risks of uncontrolled spread and the emergence of novel pathogens or resistant strains. Regulatory oversight in this field is stringent to prevent such outcomes. Correct approach analysis. The correct approach involves a comprehensive risk assessment that meticulously evaluates the potential for horizontal gene transfer, the fitness cost of the engineered plasmid to the host organism, and the environmental persistence and impact of both the host and the plasmid. This assessment must be guided by established biosafety guidelines and regulatory frameworks, such as those outlined by the relevant national biosafety authorities, e.g., the Health and Safety Executive, HSE in the UK, or the relevant bodies under the US NIH guidelines for research involving recombinant or synthetic nucleic acid molecules, if applicable to the exam's specified jurisdiction. It requires rigorous containment strategies, including physical and biological containment measures, to prevent accidental release. Furthermore, ongoing monitoring for unintended genetic changes or spread is crucial. This approach represents best professional practice because it prioritizes public health and environmental safety by adhering to the precautionary principle and robust risk management protocols mandated by regulatory bodies. Incorrect approaches analysis. One incorrect approach involves proceeding with the large-scale deployment of the engineered organism without adequate containment or risk assessment, assuming that the plasmid's intended function will be the only outcome. This fails to acknowledge the dynamic nature of bacterial genetics and the well-documented phenomenon of horizontal gene transfer, which could lead to the unintended spread of resistance or virulence genes to other organisms, posing a significant public health and ecological risk. This violates biosafety regulations that mandate risk assessment and containment for genetically modified organisms. Another incorrect approach is to rely solely on the theoretical stability of the plasmid within the laboratory setting, without considering its behavior in a complex, natural environment. Plasmids can be lost or transferred under varying environmental pressures, and their presence can confer fitness advantages or disadvantages that are context-dependent. This oversight ignores the regulatory requirement for assessing environmental impact and potential for unintended dissemination. A third incorrect approach is to prioritize the speed of research or application over thorough safety evaluations, leading to premature release or inadequate containment. This demonstrates a disregard for ethical responsibilities and regulatory mandates designed to protect society and the environment from potential harm arising from the misuse or uncontrolled spread of genetically modified biological agents. Such an approach could lead to severe regulatory penalties and reputational damage. Professional reasoning. Professionals in this field must adopt a systematic decision-making process that begins with a thorough understanding of the relevant regulatory framework and biosafety guidelines. This involves identifying potential hazards associated with the specific plasmid and host organism, assessing the likelihood and severity of these hazards, and implementing appropriate control measures. A critical step is to consult with biosafety officers and regulatory experts to ensure compliance. Continuous evaluation and adaptation of containment strategies based on new data or evolving understanding of the risks are essential. The decision-making process should always prioritize safety, ethical considerations, and regulatory compliance over expediency. Question 8. The investigation demonstrates the isolation of a novel bacterial strain exhibiting an unusual resistance profile to a panel of antibiotics. Subsequent preliminary phenotypic testing suggests a potential novel mechanism of resistance. What is the most appropriate next step to scientifically and regulatorily validate these findings within the UK regulatory framework for microbiological research? Option A. Share the raw phenotypic resistance data with a commercial diagnostic company for their interpretation and potential development of a diagnostic assay. Option B. focus on further phenotypic characterization of the resistance mechanism using biochemical assays, assuming the genetic basis will be implicitly understood from the observed biochemical changes. Option C. Conduct comprehensive genomic sequencing of the isolate to identify the genes responsible for the observed antibiotic resistance, followed by bioinformatic analysis to compare these genes with known resistance determinants in public databases. Option D. Immediately publish the preliminary phenotypic findings in a peer-reviewed journal, highlighting the potential for a novel resistance mechanism to alert the scientific community. The correct answer is C. Conduct comprehensive genomic sequencing of the isolate to identify the genes responsible for the observed antibiotic resistance, followed by bioinformatic analysis to compare these genes with known resistance determinants in public databases. Explanation: the investigation demonstrates a scenario where a novel microbial strain exhibiting unusual genetic characteristics has been isolated. The professional challenge lies in accurately characterizing this strain's genetic makeup and its potential implications, while strictly adhering to the regulatory framework governing microbiological research and diagnostics within the specified jurisdiction, assuming UK/CISI for this example. This requires a deep understanding of microbial genetics, relevant legislation, and ethical considerations for handling potentially novel or pathogenic organisms. The correct approach represents best professional practice because it prioritizes a systematic, evidence-based, and compliant method for genetic characterization. This involves employing validated molecular techniques to identify the genetic basis of the observed phenotype, followed by rigorous comparison against established databases and literature. Crucially, it mandates adherence to biosafety protocols and reporting requirements as stipulated by UK Health and Safety Executive (HSE) guidelines and relevant professional bodies like the Institute of Biomedical Science (IBIMS). This ensures the safety of personnel, the public, and the environment, while also contributing to the scientific record in a responsible and verifiable manner. An incorrect approach that bypasses systematic genetic analysis and relies solely on phenotypic observation would be a significant regulatory and ethical failure. This neglects the fundamental principle of genetic confirmation, which is essential for accurate identification and risk assessment. It could lead to misidentification, inappropriate containment measures, and potentially the dissemination of uncharacterized biological agents, violating the spirit and letter of biosafety regulations. Another incorrect approach that involves sharing preliminary, unvalidated genetic data with external parties without proper internal review or adherence to data protection regulations would also constitute a serious failure. This could compromise intellectual property, lead to premature or inaccurate conclusions being drawn by others, and potentially violate confidentiality agreements or data privacy laws. A further incorrect approach that involves extrapolating the genetic findings to predict pathogenicity without robust experimental validation and comparative genomics would be scientifically unsound and ethically questionable. This could lead to unwarranted public alarm or complacency, depending on the nature of the extrapolation, and would fail to meet the standards of evidence-based scientific reporting required by regulatory bodies. The professional reasoning process for similar situations should involve a structured approach. First, clearly define the research question and objectives. Second, identify all relevant regulatory requirements and ethical guidelines applicable to the specific jurisdiction and the nature of the work. Third, select appropriate, validated methodologies for investigation. Fourth, conduct thorough data analysis and interpretation, comparing findings against established benchmarks. Fifth, ensure all findings are documented meticulously and reported according to established protocols and regulatory mandates. Finally, seek expert consultation when encountering novel or ambiguous results. Question 9. Upon reviewing the metabolic capabilities of a novel bacterial isolate from a deep-sea hydrothermal vent, which comparative physiological approach would most accurately elucidate its unique energy generation strategies and carbon assimilation pathways, considering the extreme environmental conditions? Option A. A comparative analysis focusing on the organism's ability to ferment common sugars and its response to a panel of antibiotic resistance markers. Option B. A comparative analysis of its flagellar structure and motility patterns compared to mesophilic bacteria known for rapid growth. Option C. A comparative analysis of its electron transport chain components and its utilization of inorganic electron donors, contrasted with known chemolithoeutrophs from similar extreme environments. Option D. A comparative analysis of its cell wall composition and its susceptibility to common bacteriophages relative to terrestrial soil bacteria. The correct answer is A. A comparative analysis focusing on the organism's ability to ferment common sugars and its response to a panel of antibiotic resistance markers. Explanation: scenario analysis. This scenario presents a professional challenge due to the critical nature of microbial physiology in diagnostic and research settings. Misinterpreting or misapplying knowledge of microbial metabolic pathways can lead to incorrect identification, flawed experimental design, and ultimately, compromised patient care or research integrity. The challenge lies in discerning the most accurate and relevant physiological characteristics for a given context, especially when dealing with closely related organisms or unusual metabolic capabilities. Careful judgment is required to select the appropriate comparative approach based on the specific goals of the analysis. Correct approach analysis. The correct approach involves a detailed comparative analysis of the metabolic pathways and energy generation strategies of the target microorganism against known reference strains or related species. This aligns with best professional practice in microbiology, as it leverages established physiological principles to differentiate and characterize microorganisms. Regulatory frameworks, such as those governing diagnostic laboratories, e.g., CLIA in the US, or equivalent UK/EU standards for clinical microbiology, implicitly require accurate identification and characterization, which are underpinned by understanding microbial physiology. Ethically, providing accurate information is paramount for patient safety and scientific advancement. This approach ensures that conclusions are based on robust scientific evidence and established physiological differences, minimizing the risk of misidentification or misinterpretation. Incorrect approaches analysis. Focusing solely on morphological characteristics without considering physiological differences is an insufficient approach. While morphology can be a starting point, many microorganisms share similar appearances, making physiological traits essential for definitive identification. Regulatory failure occurs because this approach does not meet the standards for accurate identification required by accrediting bodies and public health regulations, which mandate more rigorous testing. Ethically, it risks misdiagnosis or incorrect research conclusions. Relying exclusively on genetic sequencing data without physiological correlation is also problematic. While genomics provides powerful insights, gene expression and metabolic capabilities can differ significantly from predicted phenotypes. Regulatory frameworks often require phenotypic confirmation for certain diagnostic tests. Ethically, presenting genetic data as definitive physiological evidence without corroboration can be misleading. Ignoring the environmental context in which the microorganism was isolated is another failure. Microbial physiology is highly adaptive and influenced by environmental factors. A comparative analysis that does not account for the likely metabolic demands of the original environment may lead to incorrect assumptions about the organism's capabilities. This can lead to regulatory noncompliance if it impacts the interpretation of environmental monitoring or food safety testing. And ethically, it compromises the scientific rigor of the analysis. Professional reasoning. Professionals should adopt a systematic, evidence-based approach. When faced with characterizing a microorganism, the process should involve 1. Defining the objective of the analysis, e.g., identification, strain typing, metabolic profiling. 2. Gathering all available data, including morphology, gram stain, biochemical tests, and genetic information. 3. Conducting a comparative physiological analysis, referencing established databases and literature, focusing on key metabolic pathways relevant to the organism's known or suspected niche. 4. Critically evaluating the consistency of all data points. And 5. Documenting the findings and the rationale for conclusions, ensuring compliance with relevant regulatory standards and ethical obligations. Question 10. Risk assessment procedures indicate that a research expedition has successfully collected microbial samples from a previously unexplored deep-sea hydrothermal vent system. These samples contain a diverse community of microorganisms adapted to high pressure, extreme temperatures, and unique chemical gradients. considering the potential for novel biological agents and the unknown ecological interactions. Which of the following represents the most responsible and ethically sound initial approach for handling and studying these samples? Option A. Prioritize the identification of known pathogenic microorganisms within the samples and disregard the study of non-pathogenic or metabolically unique organisms until potential threats are fully understood. Option B. Immediately initiate broad-spectrum culturing of all collected samples in a standard laboratory setting to identify and isolate novel species for further study and potential biotechnological applications. Option C. Assume that microorganisms from such an extreme and isolated environment pose no significant risk and proceed with direct analysis and potential dissemination of findings without extensive containment measures. Option D. Establish strict containment protocols within a high-containment biosafety laboratory, conduct thorough genomic and metabolic characterization of isolates, and perform risk assessments before any further experimental manipulation or potential release. The correct answer is B. Immediately initiate broad-spectrum culturing of all collected samples in a standard laboratory setting to identify and isolate novel species for further study and potential biotechnological applications. Explanation. Scenario analysis. This scenario presents a professional challenge due to the inherent variability and potential for unknown microbial life within a novel deep-sea hydrothermal vent ecosystem. The primary difficulty lies in predicting the metabolic capabilities and potential pathogenicity of microorganisms that have evolved in extreme conditions, isolated from typical terrestrial or aquatic environments. This necessitates a cautious and evidence-based approach to sample handling and analysis, balancing the need for scientific discovery with the imperative to prevent unintended environmental release or exposure. Correct approach analysis. The correct approach prioritizes containment and rigorous characterization before any potential release or broader application. This aligns with the precautionary principle often embedded in biosafety regulations, which mandates taking preventive action in the face of uncertainty. By establishing a controlled laboratory environment and conducting comprehensive genomic and metabolic profiling, microbiologists can assess risks associated with novel organisms, including their potential for pathogenicity, environmental impact, and interaction with existing ecosystems. This methodical process ensures that any subsequent research or application is informed by a thorough understanding of the organism's characteristics, thereby minimizing unforeseen risks and adhering to ethical research practices. Incorrect approaches analysis. One incorrect approach involves immediate culturing and broad dissemination of isolates without prior containment or characterization. This fails to acknowledge the potential for novel pathogens or organisms with disruptive ecological roles. Ethically, it breaches the duty of care to prevent harm to human health and the environment. Regulatory frameworks, even in the absence of specific legislation for every novel organism, generally require a risk-based approach to biosafety, which this bypasses. Another incorrect approach is to assume that because the habitat is extreme and remote, the microorganisms pose no threat. This is a dangerous assumption that ignores the evolutionary adaptability of microbes. It represents a failure in risk assessment and a disregard for the potential for unforeseen consequences, which is a violation of professional responsibility and potentially regulatory guidelines that mandate thorough risk evaluation for any biological agent. Third incorrect approach is to focus solely on identifying known pathogens and disregard other novel metabolic pathways or genetic elements. This is insufficient because even non-pathogenic organisms can have significant environmental impacts or possess novel biotechnological potential that requires careful assessment before widespread use. It represents a narrow and incomplete risk assessment, failing to consider the full spectrum of potential implication. Professional reasoning. Professionals should adopt a tiered approach to risk assessment for novel microbial habitats. This involves 1. Initial risk assessment based on habitat characteristics and known microbial ecology of similar environments. 2. Strict containment of all samples and isolates in appropriate biosafety level laboratories. 3. Comprehensive characterization, including genomic sequencing, metabolic profiling, and assessment of potential pathogenicity or environmental impact. 4. Gradual de-escalation of containment and controlled experimental progression based on accumulating risk data. 5. Adherence to all relevant national and international biosafety guidelines and regulations, even if specific organisms are not explicitly listed. This systematic process ensures that scientific advancement is pursued responsibly and ethically. Question 11. Examination of the data shows a significant shift in the microbial community composition in a freshwater lake sediment sample, with an observed increase in anaerobic sulfate-reducing bacteria and a decrease in aerobic heterotrophic bacteria. The water quality parameters indicate slightly elevated levels of organic pollutants. Considering the role of microbes in soil and water ecosystems, which of the following interpretations best reflects the likely ecological implications and requires further investigation under relevant environmental protection guidelines? Option A. The presence of sulfate-reducing bacteria is a natural phenomenon in all aquatic sediments and requires no further attention as long as organic pollutants are present. Option B. The observed shift suggests an increasing anaerobic environment due to organic matter decomposition, potentially leading to reduced oxygen availability and altered nutrient cycling, which warrants further monitoring for signs of eutrophication and potential impacts on benthic organisms. Option C. The decrease in aerobic heterotrophic bacteria signifies a complete loss of microbial function in the sediment, rendering the ecosystem inert and incapable of supporting any life. Option D. The increase in sulfate-reducing bacteria is a direct indicator of a severe contamination event requiring immediate remediation efforts to remove all microbial life from the sediment. The correct answer is B. The observed shift suggests an increasing anaerobic environment due to organic matter decomposition, potentially leading to reduced oxygen availability and altered nutrient cycling, which warrants further monitoring for signs of eutrophication and potential impacts on benthic organisms. Explanation. Scenario analysis. This scenario presents a professional challenge due to the inherent complexity of soil and water ecosystems and the potential for microbial activities to have widespread, often unseen, impacts. The challenge lies in accurately interpreting complex microbiological data within the context of environmental regulations and scientific best practices. Professionals must exercise careful judgment to distinguish between normal ecological processes and those that may indicate environmental degradation or pose a risk, requiring a nuanced understanding of microbial roles and their implications for ecosystem health and regulatory compliance. Correct approach analysis. The correct approach involves a comprehensive assessment of microbial community structure and function in relation to established environmental quality parameters and relevant regulatory guidelines. This approach is best professional practice because it directly addresses the core of the problem, understanding the ecological role of microbes in soil and water. By analyzing both the composition, who is there, and the activity, what they are doing of microbial communities, and comparing these findings against established benchmarks, a robust and scientifically defensible conclusion can be drawn. This aligns with the principles of environmental stewardship and regulatory compliance, ensuring that any observed microbial phenomena are evaluated against objective criteria for potential environmental impact. Focus on functional indicators, such as nutrient cycling or pollutant degradation, provides direct evidence of the ecosystem's health and the microbe's contribution to it, which is crucial for informed decision-making and regulatory reporting. Incorrect approach is analysis. One incorrect approach would be to focus solely on the presence of specific microbial species without considering their abundance, activity, or the overall ecological context. This is a regulatory and ethical failure because it can lead to misinterpretations. For instance, the presence of a microorganism often associated with pollution might be benign in a different environmental setting or at very low concentrations, or it might be a native species playing a beneficial role. Without functional data and contextual analysis, such an approach risks unnecessary alarm or overlooking genuine issues. Another incorrect approach would be to rely on anecdotal evidence or outdated literature without validating it against current, site-specific data and contemporary regulatory standards. This represents a failure to adhere to scientific rigor and current best practices. Environmental conditions and microbial populations can change, and regulations are updated. Basing conclusions on outdated information can lead to non-compliance with current environmental protection laws and a misrepresentation of the actual ecological state. A third incorrect approach would be to overemphasize the potential for pathogenicity without sufficient evidence or a clear link to human or animal health risks within the defined ecosystem. While some microbes are pathogenic, many are not, and their presence in soil and water is a natural part of ecosystem function. This approach fails to differentiate between ecological roles and health hazards, potentially leading to misallocation of resources and unwarranted public concern, and it may not align with the specific regulatory framework governing environmental microbiology assessments. Professional reasoning. Professionals should adopt a systematic, data-driven approach. This involves 1. Clearly defining the scope of the investigation based on the environmental setting and potential concerns. 2. Employing appropriate sampling and analytical techniques to characterize microbial communities and their functions. 3. Comparing the obtained data against relevant regulatory standards, environmental quality guidelines, and scientific literature. 4. Integrating findings from different analyses, e.g., community structure, metabolic activity, environmental parameters, to form a holistic understanding. 5. Documenting all findings, methodologies, and conclusions transparently, ensuring that any recommendations are evidence-based and aligned with regulatory requirements and ethical responsibilities. Question 12. Performance analysis shows a potential microbial contamination event in a sterile pharmaceutical product manufacturing line. While the primary process is designed to be aerobic, environmental monitoring has detected low levels of microbial growth. A risk assessment is required to determine the appropriate response. Which of the following approaches best addresses the potential risks associated with microbial metabolism in this scenario? Option A. Dismiss the detected microbial growth as insignificant, given that the primary manufacturing process is aerobic and the levels are low, without further characterization of the specific organisms. Option B. Conduct a general assessment of microbial contamination without specifically differentiating between aerobic and anaerobic metabolic pathways, relying on broad-spectrum antimicrobial treatments. Option C. Prioritize the identification and risk assessment of all detected microorganisms, considering their aerobic and anaerobic metabolic potential and their implications for product integrity and patient safety, in accordance with regulatory guidelines for contamination control. Option D. Focus solely on the identification and control of aerobic microorganisms, assuming that anaerobic organisms are unlikely to pose a significant threat in an otherwise aerobic manufacturing environment. The correct answer is C. Prioritize the identification and risk assessment of all detected microorganisms, considering their aerobic and anaerobic metabolic potential and their implications for product integrity and patient safety, in accordance with regulatory guidelines for contamination control. Explanation. Scenario analysis. This scenario is professionally challenging because it requires a microbiologist to assess the risk associated with a potential contamination event in a pharmaceutical manufacturing process. The core of the challenge lies in understanding the metabolic differences between aerobic and anaerobic microorganisms and how these differences impact their survival, growth, and potential to produce harmful byproducts under specific environmental conditions. A misjudgment could lead to the release of a contaminated product, posing a significant risk to public health and resulting in severe regulatory penalties. The need for precise risk assessment, grounded in scientific understanding and regulatory compliance, is paramount. Correct approach analysis. The correct approach involves a thorough risk assessment that prioritizes the identification and characterization of any detected microorganisms. This includes determining their metabolic capabilities, aerobic vs. anaerobic, and assessing their potential to proliferate in the specific manufacturing environment, which may have varying oxygen levels. Understanding that anaerobic organisms can thrive in low oxygen or oxygen-free environments, and that some can produce potent toxins or degrade essential product components, is crucial. This approach aligns with good manufacturing practices, GMP, and regulatory guidelines that mandate robust environmental monitoring and contamination control strategies to ensure product safety and efficacy. It reflects a proactive and scientifically sound method for mitigating risk. Incorrect approach is analysis, an approach that dismisses the significance of anaerobic microorganisms simply because the primary process is assumed to be aerobic, is a critical regulatory and ethical failure. This overlooks the reality that manufacturing environments are rarely perfectly aerobic, and that anaerobic organisms can be introduced through raw materials, personnel, or equipment. Failure to consider their metabolic potential under localized anaerobic conditions could lead to undetected contamination and subsequent product failure. Another incorrect approach is to focus solely on the identification of aerobic organisms while neglecting the potential for anaerobic contamination. This is a failure to conduct a comprehensive risk assessment. Regulatory bodies expect a thorough evaluation of all potential microbial threats, regardless of their primary metabolic preference, especially when those threats can impact product integrity or patient safety. Finally, an approach that relies on general assumptions about microbial behavior without specific testing or characterization of the detected isolates is also professionally unacceptable. This lacks the scientific rigor required for pharmaceutical manufacturing and violates the principle of evidence-based decision-making mandated by regulatory frameworks. It fails to adequately address the specific risks posed by the actual microbial contaminants present. Professional reasoning. Professionals in this field must adopt a systematic and evidence-based approach to risk assessment. This involves 1. Recognizing the potential for diverse microbial contamination, including both aerobic and anaerobic organisms. 2. Employing appropriate sampling and testing methodologies to detect and identify all relevant microorganisms. 3. Characterizing the metabolic capabilities of detected isolates and assessing their potential impact within the specific manufacturing context. 4. Implementing control measures proportionate to the identified risks, in strict adherence to all applicable regulatory guidelines and ethical obligations to protect public health. Question 13. Benchmark analysis indicates that a diagnostic microbiology laboratory is experiencing challenges in consistently isolating a range of clinically significant bacteria from diverse patient specimens. The laboratory is considering revising its primary culture media strategy to improve recovery rates, particularly for organisms with complex nutritional needs. Which of the following approaches best aligns with best professional practice and regulatory expectations for ensuring accurate and reliable microbial detection? Option A. Transitioning exclusively to the most expensive, highly enriched media available for all specimen types to maximize the chances of growing any microorganism. Option B. Implementing a standardized, enriched basal medium supplemented with specific growth factors known to support fastidious organisms, alongside general-purpose media for routine screening. Option C. Relying solely on a basic, inexpensive general-purpose agar that is widely available but lacks specific nutrient enrichment. Option D. Utilizing a single, general-purpose medium that has not been specifically validated for the variety of clinical specimen types processed by the laboratory. The correct answer is B. Implementing a standardized, enriched basal medium supplemented with specific growth factors known to support fastidious organisms, alongside general-purpose media for routine screening. Explanation. Scenario analysis. This scenario presents a professional challenge in a diagnostic microbiology laboratory setting, specifically concerning the nutritional requirements of microorganisms. The challenge lies in ensuring that the chosen culture media adequately support the growth of a broad range of clinically relevant pathogens, including fastidious organisms, while also being cost-effective and readily available. Failure to provide optimal nutritional conditions can lead to false negative results, delayed diagnoses, and inappropriate patient management, impacting patient outcomes and potentially leading to antimicrobial resistance issues due to empirical treatment. The laboratory must balance scientific accuracy with operational efficiency and regulatory compliance. Correct approach analysis. The correct approach involves selecting a basal culture medium that is known to support the growth of a wide variety of microorganisms, including those with complex nutritional needs. This basal medium should then be supplemented with specific growth factors, such as blood, serum, or enriched nutrients, to cater to the requirements of fastidious organisms. This strategy ensures that the medium is versatile enough to detect a broad spectrum of potential pathogens. This aligns with best practices in clinical microbiology, emphasizing the need for sensitive and specific diagnostic methods. Regulatory guidelines, such as those from the UK's Health and Safety Executive, HSE, and the Medicines and Healthcare Products Regulatory Agency, MHRA, implicitly require laboratories to use validated and appropriate methods that ensure accurate detection of pathogens, thereby safeguarding public health. The principle of fitness for purpose is paramount, meaning the chosen media must be demonstrably capable of supporting the growth of target organisms under the laboratory's operating conditions. Incorrect approaches analysis. One incorrect approach is to solely rely on a general-purpose medium that is inexpensive and widely available, but lacks the necessary enriched components to support the growth of fastidious bacteria. This would lead to a failure to detect organisms with specific nutritional requirements, resulting in misdiagnoses and potentially serious patient harm. This approach violates the implicit regulatory expectation of using appropriate diagnostic tools that are sensitive enough for clinical purposes. Another incorrect approach is to exclusively use highly specialized and expensive enriched media for all samples, regardless of the suspected pathogen. While this might ensure the growth of even the most demanding organisms, it is operationally inefficient, costly, and may not be necessary for the majority of routine diagnostic work. This approach fails to demonstrate prudent resource management, which is an underlying ethical consideration in healthcare provision, and could be seen as not adhering to the principle of providing effective and efficient care as expected by regulatory bodies overseeing healthcare services. A third incorrect approach is to use a medium that has not been validated for the specific types of clinical specimens being processed. Different specimen types may contain inhibitory substances or require different nutritional profiles for optimal microbial recovery. Failure to validate media for specific specimen types can lead to unreliable results, contravening the regulatory requirement for quality assurance and the use of validated laboratory procedures. Professional reasoning. Professionals in diagnostic microbiology should adopt a systematic approach to media selection. This involves understanding the common and uncommon pathogens associated with the types of specimens received, their known nutritional requirements, and the limitations of various culture media. A risk-based assessment should be performed, considering the potential clinical impact of missing specific pathogens. Consultation with clinical microbiologists and reference laboratories, as well as adherence to established laboratory protocols and guidelines, e.g. from the UK Health Security Agency (UKSA), is crucial. The decision-making process should prioritize patient safety and diagnostic accuracy, while also considering cost-effectiveness and operational feasibility. Regular review and validation of media performance are essential components of a robust quality management system, ensuring ongoing compliance with regulatory expectations. Question 14. Analysis of a situation where a specific strain of bacteria is observed to be colonizing the vascular tissues of a host plant, consuming plant sap for nutrients and causing wilting and reduced growth in the plant. Based on these observations, which symbiotic relationship is most accurately described? Option A, mutualism. Option B, predation. Option C, parasitism. Option D, commensalism. The correct answer is C, parasitism. Explanation. Scenario analysis. This scenario presents a common challenge in applied microbiology, where understanding the ecological relationships between organisms is crucial for effective control or utilization. The professional challenge lies in accurately identifying the symbiotic relationship to implement the most appropriate and scientifically sound intervention strategy. This identification can lead to ineffective treatments, unintended ecological consequences, or missed opportunities for beneficial applications. Careful judgment is required to distinguish between the subtle differences in the impact each organism has on the other. Correct approach analysis. The correct approach involves a thorough assessment of the interactions to determine if both organisms benefit, mutualism. One benefits and the other is unaffected, commensalism, or one benefits at the expense of the other, parasitism. In this case, identifying that the bacteria are deriving nutrients from the host plant while causing visible damage to its tissues directly points to a parasitic relationship. This accurate classification is essential for applying targeted control measures, such as specific antimicrobial agents or horticultural practices that disrupt the parasitic cycle, aligning with best practices in plant pathology and microbial management. Incorrect approaches analysis. One incorrect approach would be to assume a mutualistic relationship. This would be a regulatory and ethical failure, as it would lead to the protection or even promotion of an organism that is actively harming the host, potentially resulting in significant crop loss or ecosystem damage. This contradicts the fundamental principles of responsible microbial management and could violate guidelines related to pest and disease control. Another incorrect approach would be to classify it as commensalism. This would be a failure because it overlooks the detrimental impact on the host plant. While the bacteria are benefiting, the host is clearly suffering harm, which is the defining characteristic of parasitism, not commensalism. This misclassification would lead to a lack of necessary intervention, allowing the harmful effects to persist and potentially worsen, contravening professional obligations to mitigate harm. Professional reasoning. Professionals should employ a systematic approach to analyzing symbiotic relationships. This involves 1. Observing and documenting the interactions between the organisms. 2. Quantifying the benefits and harms to each organism involved. 3. Consulting established scientific literature and taxonomic classifications for similar interactions. 4. Considering the ecological context and potential consequences of each type of relationship. 5. Applying the most accurate classification to inform decision-making regarding intervention or management strategies, ensuring adherence to scientific principles and ethical responsibilities. Question 15. Stakeholder feedback indicates a growing demand for rapid identification of microbial metabolic pathway disruptions in critically ill patients to guide immediate therapeutic interventions. A new, unvalidated rapid assay for detecting key glycolytic intermediates has been developed, showing promising preliminary results, but lacking full validation against established gold standards. The clinical team is requesting its immediate use for a patient with a suspected severe metabolic disorder, citing the critical time window for intervention. As the specialist in microbiology, you are responsible for ensuring the accuracy and reliability of all diagnostic tests performed. What is the most appropriate course of action? Option A. Utilize the new, unvalidated assay and report the results, but clearly caveat them as preliminary and subject to confirmation by validated methods, while simultaneously initiating the full validation process for the new assay. Option B. Refuse to use the unvalidated assay and proceed with the established, validated methods for metabolic pathway analysis, communicating the expected turnaround time to the clinical team. Option C. Use the new, unvalidated assay and report the preliminary findings as definitive, without mentioning the lack of full validation, to avoid causing alarm to the clinical team. Option D. Implement the new, unvalidated assay immediately to provide the fastest possible results, assuming the preliminary data is sufficient for clinical decision-making in this urgent situation. The correct answer is B. Refuse to use the unvalidated assay and proceed with the established, validated methods for metabolic pathway analysis, communicating the expected turnaround time to the clinical team. Explanation. This scenario presents a professional challenge due to the inherent conflict between the need for rapid diagnostic results and the ethical imperative to ensure the accuracy and reliability of those results, especially when they have direct implications for patient care and resource allocation. The microbiologist must balance the urgency of the situation with the scientific rigor required for valid testing. The correct approach represents best professional practice because it prioritizes scientific integrity and patient safety above all else. Adhering to established protocols for metabolic pathway analysis, even when faced with time constraints, ensures that the data generated is accurate and defensible. This aligns with the fundamental ethical principles of beneficence, acting in the best interest of the patient, and non-maleficence, avoiding harm, as inaccurate results could lead to inappropriate treatment or unnecessary use of resources. Furthermore, it upholds professional accountability and the reputation of the laboratory by demonstrating a commitment to quality assurance. An incorrect approach that prioritizes speed over validation would represent a significant regulatory and ethical failure. Specifically, it would violate guidelines that mandate the use of validated methods and quality control procedures for diagnostic testing. This could lead to misdiagnosis, delayed or incorrect treatment, and potentially harm to patients. Such an approach also undermines the trust placed in the laboratory and the microbiologist by healthcare providers and the public. Another incorrect approach, such as selectively reporting preliminary findings without full confirmation, also fails to meet regulatory standards for reporting diagnostic results. This can create confusion and lead to premature clinical decisions based on incomplete or potentially erroneous information, violating principles of transparency and accuracy in reporting. The professional decision-making process in such situations should involve a clear understanding of regulatory requirements for diagnostic testing, including validation, quality control, and reporting standards. When faced with conflicting pressures, a microbiologist should first assess the potential impact of any deviation from standard procedures on patient safety and diagnostic accuracy. If time is a critical factor, the professional should explore options for expediting the validated process or communicating the limitations of any preliminary data clearly and transparently to the requesting clinician, rather than compromising the integrity of the testing itself. Escalating concerns about resource limitations or unrealistic turnaround time expectations to laboratory management is also a crucial step in ensuring appropriate operational adjustments are made without sacrificing quality. Question 16. The monitoring system demonstrates an unusual aggregation of electron-dense material within the cytoplasm of several bacterial isolates from a patient sample, distinct from typical ribosomal distribution and appearing larger than common inclusion bodies. The SM is tasked with identifying the nature of these structures. Which of the following represents the most ethically and professionally sound course of action? Option A. Attempt to identify the material based on a quick search of online databases for similar images, and if a match is found, report it as a confirmed finding without independent verification or consultation. Option B. Disregard the observation as a potential artifact of sample preparation or staining, assuming it does not represent a clinically significant finding, and proceed with routine identification of known pathogens. Option C. Immediately classify the material as a novel inclusion body and report it as a potential indicator of a rare infection, initiating further specialized testing without further consultation. Option D. Document the observation meticulously, including size, density, and location relative to the nucleoid and ribosomes, and consult with a senior microbiologist or pathologist for expert interpretation before proceeding with further diagnostic steps. The correct answer is C. Immediately classify the material as a novel inclusion body and report it as a potential indicator of a rare infection, initiating further specialized testing without further consultation. Explanation. Scenario analysis. This scenario presents an ethical dilemma for a specialist in microbiology, SM, working within a regulated environment. The challenge lies in balancing the immediate need for accurate diagnostic results with the potential for misinterpreting subtle cellular abnormalities that could have significant implications for patient care and public health. The SM must exercise professional judgment, relying on established protocols and ethical guidelines to ensure the integrity of their work and avoid potential harm. The presence of unusual structures within the cytoplasm of bacteria, even if not immediately identifiable as pathogenic, requires careful consideration and adherence to established diagnostic procedures. Correct approach analysis. The correct approach involves meticulous observation, accurate documentation, and consultation with senior colleagues or supervisors. This aligns with the ethical principles of competence, integrity, and professional responsibility expected of an SM. By thoroughly documenting the observed cytoplasmic structures and seeking expert opinion, the SM ensures that the findings are interpreted within the correct diagnostic context, minimizing the risk of misdiagnosis or overlooking a potentially significant anomaly. This approach upholds the standards of good laboratory practice and patient safety, which are paramount in a regulated healthcare setting. Adherence to established protocols for identifying and reporting cellular components, including inclusion bodies and nucleoid regions, is critical. Incorrect approaches analysis. One incorrect approach would be to dismiss the unusual cytoplasmic structures as artifacts or insignificant without proper investigation. This demonstrates a failure in competence and diligence, potentially leading to a misdiagnosis of a significant bacterial condition. It violates the ethical obligation to provide accurate and thorough diagnostic services. Another incorrect approach would be to prematurely identify the structures as a specific pathogen or condition without sufficient evidence or confirmation. This exhibits a lack of professional restraint and could lead to inappropriate treatment decisions, patient harm, and a breach of professional integrity. It bypasses the established diagnostic pathways and the need for confirmatory testing. A third incorrect approach would be to ignore the observation altogether, perhaps due to time pressure or a lack of immediate understanding. This represents a dereliction of duty and a failure to uphold the professional standards of an SM. It compromises patient care and the reliability of laboratory results, which are essential for effective healthcare. Professional reasoning. Professionals facing such situations should employ a systematic decision-making process. This involves 1. Observation and data collection. Carefully observe and document all relevant findings, including the morphology and location of cytoplasmic structures. 2. Protocol adherence. Consult and follow established laboratory protocols for bacterial identification and characterization, paying attention to guidelines on reporting unusual findings. 3. Knowledge application. Utilize existing knowledge of bacterial cytoplasmic structures, ribosomes, nucleoid inclusion bodies, to inform initial interpretations. 4. Consultation and escalation. If uncertainties arise or unusual findings are noted, consult with more experienced colleagues, supervisors, or relevant experts. 5. Documentation and reporting. Ensure all observations, interpretations, and consultations are meticulously documented in accordance with laboratory and regulatory requirements. This structured approach ensures that decisions are evidence-based, ethically sound, and professionally defensible. Question 17. Research into the microbial diversity and community structure of a novel industrial wastewater effluent has identified a significant abundance of previously uncharacterized bacterial species alongside known opportunistic pathogens. Which of the following approaches best represents a robust risk assessment strategy for this effluent? Option A. Conduct a comprehensive risk assessment that evaluates the potential pathogenicity of identified species, their ecological roles within the effluent community, potential routes of exposure to humans and the environment, and the likelihood of adverse outcomes, considering both known pathogens and novel organisms. Option B. Prioritize the identification and characterization of all novel bacterial species, assuming that their unknown nature inherently poses the highest risk, and recommend extensive containment measures. Option C. Focus solely on the presence and abundance of the known opportunistic pathogens, implementing control measures based on established guidelines for these specific organisms. Option D. Rely on historical data from similar industrial wastewater effluents to infer the risk, without conducting specific analyses of the current microbial community structure and its unique characteristics. The correct answer is A. Conduct a comprehensive risk assessment that evaluates the potential pathogenicity of identified species, their ecological roles within the effluent community, potential routes of exposure to humans and the environment, and the likelihood of adverse outcomes, considering both known pathogens and novel organisms. Explanation. Scenario analysis. This scenario presents a professional challenge due to the inherent variability and complexity of microbial communities. Accurately assessing the risk posed by these diverse populations requires a nuanced understanding of their ecological roles, potential pathogenicity, and environmental interactions. The challenge lies in moving beyond simple identification to a functional and risk-based interpretation, especially when dealing with novel or poorly characterized microorganisms. Careful judgment is required to balance the need for comprehensive data with the practicalities of risk assessment and resource allocation. Correct approach analysis. The correct approach represents best professional practice because it prioritizes a comprehensive, multifaceted risk assessment that considers both the presence of microorganisms and their potential impact. This aligns with regulatory frameworks that emphasize proactive identification of hazards and evaluation of risks. By incorporating factors such as known pathogenicity, environmental context, and potential for transmission or adverse effects, this approach ensures that interventions are targeted and proportionate to the actual risk. This is ethically sound as it prioritizes public and environmental health by addressing potential threats effectively. Incorrect approaches analysis. One incorrect approach fails by solely focusing on the presence of known pathogens without considering the broader community structure or environmental context. This is a regulatory failure because it neglects the potential for synergistic interactions within the microbial community that could either mitigate or exacerbate risk. Ethically, it is insufficient as it may lead to over- or under-intervention based on an incomplete picture. Another incorrect approach is flawed by prioritizing the novelty of identified microorganisms over their potential impact. This is a professional and ethical failure because the mere presence of a novel organism does not automatically equate to a significant risk. Regulatory frameworks typically require evidence of harm or a clear pathway to harm before mandating specific actions. Focusing on novelty without risk assessment can lead to misallocation of resources and unnecessary alarm. A further incorrect approach is inadequate because it relies solely on historical data without accounting for dynamic changes in microbial diversity and community structure. Microbial ecosystems are not static. Environmental shifts, introduction of new species, or changes in host physiology can alter community composition and risk profiles. Regulatory compliance often necessitates adaptive risk management strategies that consider current conditions, making a purely historical approach a failure to meet evolving standards and best practices. Professional reasoning. Professionals should employ a systematic risk assessment framework that begins with hazard identification, what microorganisms are present, and what are their known or potential properties. This is followed by exposure assessment. How might humans or the environment come into contact with these microorganisms? Subsequently, risk characterization integrates hazard and exposure information to estimate the likelihood and severity of adverse effects. Finally, risk management involves developing and implementing appropriate control measures. This iterative process, informed by current scientific understanding and regulatory guidance, ensures that decisions are evidence-based, proportionate, and ethically defensible. Question 18. The monitoring system demonstrates that a specific bacterial culture, intended for a quality control assay, is exhibiting significantly reduced turbidity and slower colony formation than expected. The incubator temperature has been stable within the specified range, and the culture medium's pH was verified prior to inoculation. However, the oxygen sensor readings are fluctuating erratically. Which of the following is the most appropriate immediate course of action to address the observed growth anomaly? Option A. Increase the incubation temperature by 2 degrees Celsius to stimulate faster growth, assuming a slight underincubation is the primary issue. Option B. Investigate and stabilize the oxygen sensor readings and associated aeration system, as fluctuating oxygen levels can critically impact bacterial growth kinetics. Option C. Re-prepare the culture medium with a fresh batch of reagents, as inconsistent medium composition is a common cause of poor microbial growth. Option D. Perform a gram stain and microscopic examination to confirm the morphology of the bacteria, as morphological changes might indicate an underlying issue. The correct answer is B. Investigate and stabilize the oxygen sensor readings and associated aeration system, as fluctuating oxygen levels can critically impact bacterial growth kinetics. Explanation. Scenario analysis. This scenario presents a professional challenge because deviations from optimal growth conditions can compromise the integrity of microbiological cultures, leading to inaccurate results, potential contamination, failure to meet regulatory standards for quality control or research. The microbiologist must exercise careful judgment to identify the root cause of the observed growth pattern and implement corrective actions that are both effective and compliant with established protocols. Correct approach analysis. The correct approach involves a systematic evaluation of the environmental parameters that directly influence microbial growth, specifically temperature, pH, and oxygen levels. This aligns with best professional practice as outlined in standard microbiological laboratory guidelines and regulatory frameworks that mandate controlled environments for microbial cultivation. By correlating the observed growth characteristics with known optima and limitations for the specific microorganism, the microbiologist can accurately diagnose the issue. This methodical approach ensures that interventions are targeted and evidence-based, minimizing the risk of introducing further variables or compromising the experiment. Adherence to these principles is crucial for maintaining data reliability and meeting the stringent requirements of regulatory bodies governing laboratory operations. Incorrect approaches analysis. One incorrect approach is to immediately adjust the temperature without considering other factors. This is a regulatory failure, because it bypasses a comprehensive assessment of all critical growth parameters. It is ethically questionable as it risks introducing a new problem or masking the true cause, potentially leading to flawed conclusions and misreporting of results. Another incorrect approach is to assume the observed growth is normal for the organism without consulting established growth curves or optimal conditions. This represents a failure to adhere to scientific rigor and quality assurance protocols. Ethically, it demonstrates a lack of diligence and could lead to the acceptance of substandard results, undermining the credibility of the laboratory. A third incorrect approach is to focus solely on the pH and ignore the oxygen requirements. This is a regulatory failure, as it neglects a key variable in microbial metabolism and growth. It is professionally unsound because it leads to an incomplete diagnosis and potentially ineffective corrective actions, failing to uphold the duty of care in ensuring accurate microbiological assessments. Professional reasoning. Professionals should employ a decision-making process that prioritizes systematic investigation and data-driven conclusions. This involves 1. Clearly defining the observed anomaly. 2. Consulting relevant scientific literature and established protocols for the specific microorganism. 3. Evaluating all potential contributing factors, temperature, pH, oxygen, media composition, etc. in a logical sequence. 4. Implementing corrective actions based on the most probable cause. 5. Verifying the effectiveness of the corrective actions through remonitoring. 6. Documenting all observations, actions, and outcomes meticulously. This structured approach ensures accountability, reproducibility, and compliance with regulatory expectations. Question 19. The performance metrics show a significant increase in the detection of heat shock protein expression in bacterial isolates from wound infections. In the context of specialist in microbiology, SM, practice, which of the following interpretations best aligns with best professional practice and regulatory expectations for reporting such findings? Option A. Interpret the heat shock protein expression as evidence of laboratory error in sample processing and initiate a full review of all recent wound infection cultures. Option B. Report the presence of heat shock protein expression as a direct indicator of impending patient treatment failure, recommending immediate escalation of antibiotic therapy. Option C. Conclude that the increased heat shock protein expression signifies a novel, highly virulent strain requiring immediate public health notification and isolation of all affected patients. Option D. Document the presence of heat shock protein expression, noting its known association with bacterial stress and potential implications for antimicrobial tolerance, and await further clinical correlation from the treating physician. The correct answer is B. Report the presence of heat shock protein expression as a direct indicator of impending patient treatment failure, recommending immediate escalation of antibiotic therapy. Explanation. Scenario analysis. This scenario presents a professional challenge in a diagnostic microbiology laboratory setting, specifically concerning the interpretation of microbial stress responses in relation to patient outcomes. The challenge lies in translating observed cellular phenomena, stress responses, into actionable clinical insights without overstepping the boundaries of diagnostic interpretation or making unsubstantiated claims. The need for careful judgment arises from the potential for misinterpretation to lead to inappropriate patient management, impacting both clinical efficacy and patient safety. The specialist in microbiology, SM, must navigate the complex interplay between fundamental microbial biology and its practical application in a regulated healthcare environment. Correct approach analysis. The correct approach represents best professional practice because it aligns with the core principles of diagnostic microbiology and regulatory compliance. It focuses on accurately identifying and reporting the presence of specific microbial stress responses that have established clinical relevance, such as those indicating potential antimicrobial resistance mechanisms or altered virulence. This approach adheres to the regulatory framework by ensuring that diagnostic reports are evidence-based, clinically relevant, and do not introduce speculative interpretations. Ethically, it prioritizes patient welfare by providing reliable information that supports informed clinical decision-making, avoiding the generation of misleading data. The emphasis on established clinical correlation ensures that the reported findings are directly applicable to patient care, as mandated by laboratory accreditation standards and professional guidelines. Incorrect approaches analysis. One incorrect approach involves broadly attributing patient treatment failure solely to the presence of any observed microbial stress response without specific, validated clinical correlation. This fails to meet regulatory requirements for diagnostic reporting, which demand evidence-based conclusions. Ethically, it is irresponsible as it could lead clinicians to make treatment decisions based on unproven associations, potentially harming patients. Another incorrect approach is to report the presence of stress responses as definitive indicators of treatment efficacy or failure without considering other contributing factors, such as host immune status or drug pharmacokinetics. This oversimplifies complex clinical scenarios and violates the principle of providing accurate and comprehensive diagnostic information. It also contravenes regulatory guidelines that emphasize the holistic assessment of patient conditions. A further incorrect approach is to recommend specific therapeutic interventions based solely on the identification of a stress response, without explicit regulatory approval or established clinical protocols. This constitutes practicing outside the scope of a diagnostic laboratory and poses significant ethical and regulatory risks, potentially leading to adverse patient events and violating professional conduct standards. Professional reasoning. Professionals in this field should adopt a decision-making process that prioritizes accuracy, clinical relevance, and regulatory compliance. This involves 1. Understanding the established clinical significance of observed microbial stress responses. 2. Consulting relevant, up-to-date scientific literature and clinical guidelines. 3. Reporting findings in a clear, concise, and unambiguous manner, explicitly stating any limitations or established correlations. 4. Collaborating with clinicians to ensure that diagnostic reports are integrated effectively into patient management plans. 5. Adhering strictly to the laboratory's quality management system and regulatory requirements for reporting and interpretation. Question 20. Which approach would be most professionally appropriate when a critical diagnostic reagent is found to be expired and there is an urgent need for patient testing? Option A. Proceed with testing without verifying the reagent's expiry status to avoid any delay. Option B. Use the expired reagent but add a disclaimer to the report indicating the reagent's expiry date. Option C. Refuse to use the expired reagent and immediately initiate the process to obtain a new, unexpired reagent, documenting the situation and the delay. Option D. Attempt to revalidate the expired reagent in-house using informal methods to see if it still performs adequately. Correct answer is C. Refuse to use the expired reagent and immediately initiate the process to obtain a new, unexpired reagent, documenting the situation and the delay. Explanation. This scenario presents a professional challenge due to the conflict between the immediate need for a potentially life-saving diagnostic test and the established protocols for ensuring the integrity and accuracy of laboratory reagents. The microbiologist is faced with a situation where deviating from standard operating procedures, SOP, could lead to inaccurate results, potentially impacting patient care, while adhering strictly might delay a critical diagnosis. The ethical imperative is to balance patient well-being with scientific rigor and regulatory compliance. The correct approach involves prioritizing patient safety and diagnostic accuracy by adhering to established quality control procedures for reagents. This aligns with the fundamental principles of good laboratory practice, GLP, and regulatory frameworks governing diagnostic laboratories, which mandate that all reagents used in patient testing must be properly validated and within their expiry dates. The specialist in microbiology, SM, exam emphasizes adherence to these standards to ensure reliable and reproducible results. By refusing to use the expired reagent and initiating the process for obtaining a new one, the microbiologist upholds the integrity of the diagnostic process, prevents potential false positives or negatives, and safeguards patient outcomes. This demonstrates a commitment to professional responsibility and ethical conduct, as mandated by the regulatory framework for diagnostic laboratories. An incorrect approach would be to use the expired reagent, even with a disclaimer. This action directly violates GLP principles and regulatory requirements for reagent quality. The expiry date signifies the point at which the manufacturer can no longer guarantee the reagent's performance characteristics, such as its specificity, sensitivity, or stability. Using an expired reagent risks generating inaccurate test results, which could lead to misdiagnosis, inappropriate treatment, or delayed necessary interventions, thereby compromising patient safety. This failure to adhere to quality control measures constitutes a significant ethical and regulatory breach. Another incorrect approach would be to bypass the quality control entirely and proceed with testing without any reagent verification. This demonstrates a disregard for established laboratory protocols and regulatory mandates designed to ensure the reliability of diagnostic testing. Such an action undermines the credibility of the laboratory and the accuracy of its results, posing a direct threat to patient care and public health. It represents a failure to uphold professional standards and a disregard for the principles of good laboratory practice. A third incorrect approach would be to attempt to revalidate the expired reagent in-house without proper validation protocols or manufacturer guidance. While a desire to resolve the reagent shortage is understandable, attempting to circumvent established validation procedures can lead to unreliable results. The integrity of diagnostic testing relies on validated reagents and methods. Unsanctioned in-house revalidation, especially for expired reagents, can introduce unknown variables and compromise the accuracy and reliability of the test results, leading to potential patient harm. This approach fails to respect the established scientific and regulatory processes for ensuring reagent quality. The professional decision-making process in such situations should involve 1. Immediate identification of the issue expired reagent. 2. Assessment of the impact on patient care and diagnostic accuracy. 3. Adherence to established SOPs and regulatory requirements regarding reagent use. 4. Communication with relevant personnel e.g. laboratory manager, procurement, to expedite the acquisition of a new reagent. And 5. Documentation of the situation and the actions taken. This systematic approach ensures that patient safety and diagnostic integrity are maintained while addressing operational challenges.